This is a mini lecture on metabolism and ATP. We're responsible for this material on the upcoming exam. Here are our goals: to talk about metabolism as a whole and what it does and why ATP is so important to it. So metabolism as a whole, we're trying to figure out how are we going to get from here to energy you can actually use. So no matter what kind of meal you like to eat, if it's healthy or if we're being more realistic and it's a Whataburger, how does that food that we eat then get turned into energy that our bodies can use for exercise, for studying, for all the things that your body needs to do? Our overall definition for metabolism is all the chemical reactions that take place in a cell. So here's a big map. It's showing everything from energy metabolism, carbohydrate metabolism, lipid metabolism, all these different kinds of pathways. These different dots are different intermediate molecules, and then there are enzymes in here that tell us how these different compounds get made and how they're used in our bodies. Two different types of metabolism that fall under that category: anabolic pathways. So these are any of the pathways that are going to build something. So they're going to make something out of subunits, out of smaller pieces, something else. These reactions are endergonic, meaning that they actually require energy. You have to put energy in to be able to make something. The other type is catabolic. These are the degradative pathways. You're degrading something, you're breaking it down. These pathways are exergonic in that they are going to release energy when they happen. So ATP is the main molecule we're going to talk about. We need to get from our food, our cheeseburger, our fruits and veggies. Our cells can't use those in their original form, but the cells can use them to make a type of energy it can use, such as ATP. So the energy from your food is used to make ATP, which your cells can then use. Cells can use ATP for things like movement, active transport, which we just talked about, or enzymatic reactions. It can help enzymes function. Not all enzymes need ATP, but many of them do for their jobs. Over here we have the structure of ATP. So here we have adenosine. You'll remember this from when we talked about nucleic acid structure. We've got a ribose sugar attached to adenine, which is one of our nitrogenous bases found in nucleic acids. So these two pieces together are called adenosine. And then we have three phosphate groups, which make up the triphosphate part. And these are the parts that are going to give this molecule the energy that it needs. Now, when we actually use this molecule for energy, what's going to happen is this bond here is going to be hydrolyzed. So it's going to use water, hydro, to break this apart, lyse, and break one of these phosphates off of it. And then what we're left with is over here. Now we have ADP, so adenosine diphosphate, di for two, and a phosphate molecule, which we often just label as PI. So when this happens, energy is released. Now, the molecule itself, ATP, we often talk about it being high energy, but what that means is breaking it is actually going to release the energy. So the molecule itself is not what we're talking about when it's high energy, but the process of breaking it apart is what's going to release the energy. And the energy is released when these new bonds are formed that produce ADP and PI. There's more energy released from doing that than what was required to actually break the bonds. So the net change in energy, you have more energy after the fact than when you started. And that's what's going to allow it to release that extra energy. Now, there are a couple different reasons that ATP has this high energy. One of those is charge repulsion. So if you look over here, we've got negative charges on each of these phosphate groups. And we know that like charges, so a negative and a negative, those repel each other. So you can imagine that if you're trying to put this molecule together, you're trying to push magnets, strong magnets together, and they just want to repel each other. So that's why it takes energy to actually put them together. But then when you break them apart, so imagine you're holding two magnets together that are trying to repel each other. When you let go, those magnets are going to spring apart and separate, and that's what's releasing energy. So taking one of these phosphates away from the others, getting this charge away from this one, is going to actually release energy. Second reason is resonance stabilization. So this is going back to your chemistry, remembering this a little bit. So in our phosphate group, look over here, we have a double bond between this phosphate and this oxygen. And this is how we represent it, but it actually isn't this way. So those electrons in that double bond, they actually pass back and forth between all of these bonds around this phosphate. So if we look over here, this is more an accurate representation of what that phosphate looks like, with that extra double bond kind of traveling around between those four. So this phosphate by itself, because it can do that, it around all four of those bonds is very stable. When we attach it to other phosphates, like we have over here in our ATP structure, now you reduce the number of oxygens that those resonance bonds can travel around, and that actually destabilizes the molecule and makes it have higher energy. So that lack of stabilization actually gives it higher energy. And the last reason is increased entropy. So this molecule, as we take it apart, now that phosphate is no longer fixed in this stable structure here. So we're increasing our entropy, increasing our randomness of those. Now we got two molecules instead of one, and that's going to release energy as well. Here we have a table of a bunch of different phosphorylated compounds that are found in the body. And the phosphate groups can be transferred back and forth between these molecules. Over here we have the amount of energy that is released from losing a phosphate from any of these. Now the general idea is that you can pass phosphates around back and forth between these compounds. However, it can only go down. So you can only pass a phosphate to something that is lower in energy, but for example, glucose 1-phosphate can't phosphorylate or can't add a phosphate to something that is higher in energy, like ATP. So ATP is here right in the middle of this chart, which is actually very important. So this means that ATP can act as a donor, so we can donate one of its phosphates down the chain to one of these other molecules, or if we have the diphosphate form here, ADP, it can actually be phosphorylated by one of these that are above it. So those can add a phosphate group to ADP. So ATP being in the middle here means it can be a donor or an acceptor of a phosphate group, and that also means it's reversible. So if we break apart an ATP, and now we have ADP, one of these other compounds can add another phosphate to make ATP again. So we can go back and forth and reuse that ADP. It's not just done once it's been changed to ADP.